Are we playing God with our genes? Gene editing and human enhancement #1 from the years 1990 to 2000. What makes you think this kid is mine? Boy, those faces look similar. A gene is a unit of inheritance. Mommies and daddies give genes to their children. As you probably know, each cell in our body contains 46 chromosomes, 23 from mommy, 23 from daddy. Well, usually nature isn't always perfect in passing these on. A chromosome is a chain of nucleotides called DNA, deoxyribonucleic acid. A chain, like a long piece of string, and it's folded so that it looks like a double helix, figure eight, something like that. A gene is simply a geographical location on each one of those chains. So a gene could be, 5,000, 25,000 base pairs long. It is just a section on the DNA that codes for proteins. That is to say, it makes our bodies. The word genome refers to the total number of genes that you and I have in our cells. So you have a genome, I have a genome. They're slightly different, to be sure. And that's why we look a little different. But we human beings share 90+%, 99+% of the genes. So our genomes are very similar, even though they're not identical. 1990 is an important year because that marked the beginning of the Human Genome Project. Oh, yes, our scientists and U.S. Congress were getting ready in the years prior. But once the legislation was passed and the funding allotted, the program began in 1990. The public program, that is, but it wasn't long, and a private program spun off. And the 1990s was like a horse race between the private and the public. Who's going to get to the finish line? The finish line consisted of winning on 2 counts. #1, sequencing the DNA. It was estimated there would be about 3 billion DNA base pairs. And then the second goal, of course, was to identify and locate the genes. Did they make it? Yes, they did. The first director of the U.S. Public Genome Project was James Watson, Nobel Prize-winning geneticist. He approved an application we submitted from the Center for Theology and Natural Sciences at the Graduate Theological Union to study the theological and ethical implications of the Human Genome Project. And here you can see Roger Shin, theologian and ethicist from Union Seminary, New York, Karen LaBox, then at Pacific School of Religion, a bioethicist. And we worked from 1990 till about 1994, monitoring the genomic research itself and then drawing off implications for theological anthropology and social ethics. As I mentioned, the Genome Project split shortly after it began. James Watson got into an argument with this man in the lower left-hand corner, Craig Venter, because Venter, a grantee, was filing for patents on DNA sequences that he had discovered, and Watson believed he ought not do that. Raw genomic knowledge should not be patentable. They had a big argument. By the time the dust settled, Watson was gone and Venter was gone. And Francis Collins there on the right-hand side took over from Watson and became the director and shepherded the government-funded Genome Project until it was completed. Venter then got private funding and entered into a horse race. Francis and Craig raced to the year 2000 to see who would get the most data about DNA sequences and the human genome. And a fascinating meeting of the AAAS, the American Association for the Advancement of Science, in February 2001. Both Francis and Craig each got an evening public forum for sharing the results of their respective genome projects. It's done. And the two projects came up with pretty much the same information, all of which was shared with the worldwide genomic community. Yeah, I was there. It was exciting. When James Watson initiated the Genome Project in the late 1980s, he estimated there would be 3 billion base pairs in the human DNA, and he also estimated there would be 100,000 genes located on those chains of DNA. When they were done, they confirmed the first one. Yes, indeed, there were 3.2 billion nucleotides. Yep, right on the money. But there was not 100,000 genes. In fact, they debated. Francis thought he found 35,000 genes, and Craig thought he found 25,000 genes, and they weren't even sure what a gene was anymore. Well, part of it was that the earlier assumption back in 1990 was that each gene would code for a single protein. Francis, on that evening of his address at the AAAS, said, well, there's a big surprise here. One gene can code for 3 or 4 different proteins, and they have to rethink what their original assumptions were. Rethinking assumptions based upon new information, that's part of the excitement of the frontier of science, especially when it's moving as fast as the frontier of human genomics has been moving since 1990 down to the present day. Now, here is Craig Venter interviewed by the San Francisco Chronicle. And this little interchange between Venter and the reporter is just the tip of a really big iceberg, and we're going to be looking at that iceberg later. So the reporter asked, "What? Only 30,000 genes? I thought there were supposed to be 100,000." So Craig Venter responds to the reporter, "Genes can't possibly explain all of what makes us what we are," said Craig Venter. Then the reporter asks rhetorically, "So how did we come to believe that our very soul is encoded in our DNA?" The reporter thought that Venter thought the human soul is encoded in the DNA. Where did that come from? What does that mean? Well, fasten your seatbelt. There'll be some twists and turns, and we're going to figure out exactly what that means. Let's add a sidebar to our story for a moment and just ask, soul in the DNA, what could that mean theologically? Well, let's go back to a conversation in the Islamic tradition. We'll start with Ibn Sina, known as Avicenna, who influenced St. Thomas Aquinas. Ibn Sina, kind of like Plato, thought of the soul as being quite independent of the body. The soul is not the form of the body, and so there's no imprint of the soul on the body, and the body does not imprint the soul. If we were to follow that tradition, there would be no interaction then between our DNA and our soul. Bioethically, you could proceed with re-engineering our genome, and we wouldn't have to worry about it affecting our soul or our relationship with God. There's another tradition beginning with Al-Ghazali in Persia, and he was more of an Aristotelian in which the soul is the form of the body. Hylomorphism is the technical term there. And the soul's activity is engaged with the physical and social nexus. So if we were to follow the Al-Ghazali tradition, we might want to think of the soul and the DNA as being connected. In fact, we might even think of the DNA, if it's the soul incarnate, is sacred. We might want to invoke the commandment, "Thou shalt not play God. Leave that soul alone." So when the reporter asked Craig Venter, "Well, wait a minute, I thought the soul and the DNA were interconnected and that we should expect a far more complex DNA configuration with 100,000 genes. What's this lousy, stinking 30,000 gene stuff about?" Well, let's return now to our story and ask ourselves the question, what do scientists think today is the number of genes in the human genome? What do genomists today think about the number of coding genes in the human genome? 21,306. Well, that's pretty precise. How about that number will change gradually over time? We think it has something to do with your genome. With regard to the role that we played here in Berkeley within the larger Human Genome Project,